Most all research demands extreme patience. Breakthroughs take time. But when it comes to learning more about genetics, Edward Marcotte was determined to speed up the process.
Part of what we do is we try to develop technologies that allow us to make measurements on thousands of genes at once. So we end up doing things like roboticizing them so that you can do a bunch at once.
As a UT Austin undergraduate, he got his first exposure to studying genetics.
Partway through college, I ended up taking a class in genetics, and I was just really amazed. It was fascinating to learn about what was known about biology, how your cells worked, and it's just absolutely amazing.
After earning his Ph.D. at the University of Texas, he spent 4 years at UCLA for his postdoctoral studies before returning to UT Austin and starting a lab in functional genomics and bioinformatics. Edward's lab enlists computer scientists, engineers, molecular biologists, and chemists. This interdisciplinary approach has produced the most complete research available today for how proteins within cells function. This new research is fundamental in understanding what different genes do.
One of the most important issues in science is asking questions and asking the right questions. And the second, perhaps most important issue, is being able to develop a plan to implement the quest for addressing these questions. And he does both.
To increase the pace of his research, Edward's lab has developed technologies enabling them to experiment on thousands of proteins and genes at a time. Working with basic genetic structures in yeast and tiny worms, they're discovering possible correlations to human genetics.
More recently, he identified a number of different genes that can cause cancers in both model organisms and also very likely in humans.
We've looked at things like predicting which genes make worms live longer if you target them with drugs. And we were able to successfully predict genes that would increase the lifespan of a worm by 50%. Again, it's not a human, it's a far cry, but humans have those genes. And so it's possible that the same thing would work in a human.
As Edward's team characterizes the function of previously unknown proteins and genes, they are better equipped to predict what happens when genes malfunction.
What I hope is that some of the things that we're developing would translate into being able to diagnose genetic cause of disease and actually be useful in that way.
By emphasizing new techniques as much as his ongoing research, Edward is substantially advancing our knowledge of genetics.
It's very satisfying to think that you could exploit your curiosity to actually do something useful.